Well, I want to thank everybody for coming. I want to thank Meg for that amazing introduction. And I can't tell you how thrilled I am to receive this award. And I hope you get the excitement through the short lecture that I'll be giving. So I want to start out and I want to introduce Earl and Thressa Stadtman. They were first-rate biochemists. First husband and wife team at the National Institutes of Health defying anti-nepotism rules. They were married for 65 years. They collaborated and had independent scientific programs. And the Stadtman way refers not only to the extraordinary high standard of rigor that they set in the biochemical research, but also their generous sharing of credit in publications with more junior scientists. And I think that these are really important things that we should all remember. And today, what I want to share with you is that we have a very interesting scientific link with both of them. So, about 15 years ago, doing unbiased basic science, which is really important, and we have to let everybody know this, we discovered something very important. And so what we did is we just followed the science. And so I'm going to take you through that path today. And it all starts with a graduate student, Melanie Yarborough, who was studying a virulence factor in Vibrio that basically, whenever it went into cells, it basically collapsed the actin cytoskeleton. So we went to the normal targets that pathogens go to, and these are Rho GTPases. And they basically are in one shape when they bind GDP, and when they bind GTP, they form a different shape. And that is like a key that fits into a lock. And in this case, it induced changes in the actin cytoskeleton. So what we noticed is that when BiP was around, the key no longer fit into the lock. And so using mass spectrometry, we figured out that the key changed shape and size, and this was by 329 Daltons. And this was quite a surprise because it didn't fit with any of the known post-translational modifications that had been discovered at the time. But then we figured out what it was, and it was an AMP. So we came up with this new term that now I think people accept called AMPylation, because it's basically an AMP modification on a protein. So basically what's happening is, you know, with a kinase, it takes ATP and it transfers the gamma phosphate to a serine, threonine, or tyrosine residue for phosphorylation. This enzyme flips that metabolite in its catalytic site and now transfers AMP to a serine, threonine, or a tyrosine residue. And so I want you to remember that BiP does this by a FicD domain. And I was really excited about this, and this was in 2008. And I'm walking down the hall, and I run into this guy, and I say, this is Mike Brown. I'm sure many of you know. Look at the smirk. And so Mike's in the hallway, and I go, Mike, I have a new post-translational modification. It's AMP on a G protein. And he was speechless, which actually never happens. But then he proceeded to tell me, as a former Stadtman postdoc, that Earl Stadtman had discovered that AMP could modify proteins. And he was involved, as you heard earlier, in studying regulation of nitrogen metabolism, and he showed that glutamine synthetase is regulated by a reversible post-translational modification with AMP on a tyrosine residue. And this was done by a nucleotidyl transferase. So this is a different domain. And so basically, over the years, what we had discovered at this time, actually at the point of this discovery, I'm showing a FicD domain, and we had solved the structure of BiP, and the structure of the nucleotide transferase had been discovered. But these are two different domains that carry out the same chemistry. So when we discovered the FicD domain, many people had studied this, including my former postdoc mentor, Jack Dixon, for many, many years, and no one knew what a FicD domain did. So they all tested whether it could do AMPylation, but then they realized not all of them carried out AMPylation. Some of them did phosphocholination, and some of them did umpylation. And there's even a FicD domain that flips the ATP in the catalytic site, and it carries out phosphorylation. So this was really, really interesting. And then I had a student, Anju Srilatha, that decided that she wanted to go do a postdoc with Vinny Tagliabracci. And he studied pseudokinases. And she walked over into my office one day and she said, Kim, I just solved the structure of my pseudokinase, and the ATP is flipped in the catalytic site, and it carries out ampylation. It's a very small world. And so then Vinny's lab went on to study other pseudokinases, and he found out they can carry out RNAylation. So instead of transferring the AMP, they transfer an RNA. So this is very significant because Anjou's kinase was cell O. And cell O is a protein that requires selenium. And it turns out Thressa Stadtman demonstrated that selenium is an essential constituent of several enzymes. So both parts of the family are part of our family for studying ampylation. And I just think this is so wonderful, the discovery that Anjou made. Now, when we're thinking about this picture, it's a very complex picture. So what I want you to realize is that this one post-translational modification is carried out by multiple folds. And I mentioned all those folds on my last slide. But the other thing that I want you to realize is a single FicD domain can accommodate a number of different metabolites. So a single fold can have diverse modifications. And what I also want you to realize is that AI cannot reveal the activity of your novel protein. In order to figure out what your protein is doing, you have to do biochemistry. And as a pertinent example of this, and I'm going to give you a beautiful example. In this paper, I must say I'm very, very proud of that Wei Peng did, and it actually went in and it required no revisions. And what he discovered is there's yet another FicD domain from a bacterial protease, a bacterial pathogen, that carries out rhamnosylation. Now, why is this important? It's UDP rhamnose. This is a sugar that's being transferred. So what he discovered was exactly what I presented on the last slide. He has a new fold for a glycosyltransferase, and we have a chapter now in Carbohydrate Biochemistry Bible for a new enzyme that carries out glycosyltransferase. So there's many things that we need to discover, and not all of them can be done in situ. So what I want to go on now is I want to talk about FicD domains in metazoans. So when we discovered the FicD domain, we realized that FicD domains were used as housekeeping proteins in bacteria. Many pathogens carry them as virulence factors, but every metazoan genome contains one gene that encodes a FicD domain. So we thought this must be important. So the day that I submitted my paper on BoP-S, I was in Switzerland with Helmut Kramer, and Helmut Kramer studies fruit flies. And I said, this is the perfect thing. I worked on fruit flies a long time ago, so we should look at this. And so I asked if we could knock out FIC in fruit flies, and we did this. And FIC null flies are viable, but FIC-mediated ampylation is required for normal visual neurotransmission in flies. So there was some kind of role that it was playing. And we were interested in this, but we were also interested in the biochemistry of this. So I had a student that was studying the metazoan FICs, and my favorite experiment for anybody to do if they're looking at a new protein is subcellular fractionation. Where's your protein expressed? And so she found that it's localized to the endoplasmic reticulum. So it's a membrane-bound protein, and she showed that the catalytic domain of FIC is in the ER. So that meant its substrate's probably in the ER. So we went to go and look at the substrate, and I was not happy about the result because I was a graduate student in Joe Sambrook and Mary Jane Gething's lab, and I swore I would never work on the molecule BiP. But it turns out that the substrate for FIC is BiP, and so now I am working on BiP and continue to work on BiP. So what is BiP? BiP is a chaperone that's found in the ER that deals with helping proteins fold. But what Halen found was that FIC modified BiP. And what I can tell you is whenever the FIC enzymes modify something, they're inactivating it. Just like we did with the Rho GTPases, BiP is inactive. So there's a pool of BiP that's inactive by ampylation. But she saw that when she stressed the cells, all of the ampylated BiP disappeared. So it is exactly this, that there's a reserve of BiP. And there's BiP that's used just, under non-stress conditions. But as soon as cells feel a stress, like a physiological stress, they release that pool of BiP by de-ampylating the BiP, and now you have more BiP to deal with stress. But we didn't know how the reaction was reversed. And so BIP is involved in the unfolded protein response, and this is a really important thing that happens in our cells. So BIP is used to help fold proteins, but if there's too many proteins that need to be folded, then you induce the UPR, the unfolded protein response. This is energetically costly for a cell, because there's RNA degradation, protein degradation, and you inhibit translation. And if you want to recover, you have to make all of those things again. Meanwhile, what the cell is doing is making more chaperone to deal with the unfolded proteins, and once they get that settled, then you can go back to homeostasis. But if you don't do that, you die. Okay, so this is a really costly thing, and it's a really important decision for the cell to make. So, but we knew we had this little rheostat that was working at the beginning of this whole UPR. And then Amanda Casey, who you'll hear more about later, joined my lab. And what she discovered is that the deampylation of BIP is by the same enzyme that ampylates BIP. So basically, when cells are not stressed, there's a pool of ampylated BIP. And when cells get stressed, FicD changes its activity to deampylate BIP, releases that BIP so it can handle with the UPR. So what I've described to you is a rheostat. So you get a little stress, you release a little BIP, you go back to homeostasis, you put more BIP that's sequestered and is not functional and is there to use when cells get stressed again. So this is the rheostat that we discovered. Now, I'll tell you right now that we don't know how it switches, and we're still working on that. We're getting close, but we haven't figured that out quite yet. But the enzyme basically has two activities, and it's all dictated by whether you're under stress in the ER or not. Okay, so I told you that we went to flies, and so this is a really, really important experiment that was done by a student in Helmut Kramer's lab, Andrew Mulham. So basically what you're looking at is the wild type and mutant fly eye. Basically, it's the rhabdomere that contains rhodopsin. And so what happens is when the flies have light, they're under a lot of stress, but that's in order for them to be able to see. And they need to recover from that stress, as do all of we, by going to sleep. Now, they don't have eyelids, so basically you turn off the lights. And so that's how they recover. So what he did is he asked, well, what happens if these eyes face physiological stress? So he had the flies under 3 days of light-dark cycle, and then he took the flies and he put them under 3 days of light with no dark. And so basically you can see the eyes get a little stressed. But then if he takes the wild type and he puts them back under 3 days of light dark, they recover. And if he does this over 3 times, you can see it's 100% recovery with the wild type. Now what happens if you no longer have that rheostat and that backup pool of BIP? So basically in the FicD nulls, if you treat them with stress, this is what their eye looks like. It doesn't look very good. And then if you get it to recover, then what happens is it recovers, but not so well. And so basically, if you basically calculate how well the eyes are functioning, as in the graph above in blue, but for the mutant, this is what it looks like. So over 3 iterations of this, you're down to 50% function, basically how the eyes are working. But what's happened is under this physiological stress is you had amplified damage, and then you had a delayed recovery. And this all aligns with increased UPR. So what Andrew discovered was using a key basic science experiment using a model organism. He did a very simple experiment to ask what's the role of FicD and what's the involvement of physiological stress. And basically what he found was FicD-mediated ampylation of BiP is important for terminally differentiated cells when dealing with physiological stress. And this is really important, the terminally differentiated, and I'll get to this in a minute. So we did all this work in flies, and I must say, the key of FicD we actually figured out in flies, but many people were like, what happens in mice? What happens in humans? You know, we need to know this. And so a lot of people weren't paying attention to our fly work. So Amanda Casey, she worked on yeast, and then she worked on the flies, and she said, well, now we need to work on mice. So what I want to tell you about Amanda is she's amazing. She's basically developed the program that I'm going to tell you about. It's hers to take with her, and she's on the job market. So if you're interested, she's in the audience, and you can talk to her after the talk. But this is absolutely amazing what she did. So she basically created the FicD null mice over COVID. And basically, the gist of many, many different experiments is that for the FicD mice faced with physiological stress, that's eating, exercising, aging, everything that we all do, we observe that many tissues, the pancreas, the muscle, and the bone, exhibit UPR-mediated damage and delayed recovery. And I want to say that this is very similar to the fly eye. For FicD liver, however, that's a regenerative tissue. So it has terminally differentiated cells, but if they get damaged, they can make new cells. We don't observe any obvious damage with repetitive stress. And FicD is important, again, for terminally differentiated cells dealing with physiological stress in the mouse model, just like the fly model. So here what I'm showing you is, did it go? Yeah. Oh, these are little mice running along. And so they're about 1 year old, and on the top is the wild type, and on the bottom is the FicD. And what you can see is that the little guy on the bottom, he's having a problem with his hind limb coordination, and that's all quantitated in the graph on your right. And so this was really interesting, and these are studies that are still ongoing with Amanda. But in the meantime, about a couple years ago, this came out. BiP inactivation due to the loss of deampylation function of FicD causes motor neuron disease. And then these same authors went and got our flies, and they basically looked at the loss of FicD causes progressive neurodegeneration in Drosophila model, our flies, of hereditary spastic paraplegia. So these are rare genetic disorders, and what I'm showing you is a motor neuron defect. But remember, I talked about different tissues. So Amanda also looked at the pancreas, and in this case, this was a pharmacological stress that induces pancreatitis with serulin, and I won't go through all the experiments except to say that it gives you the same type of phenotype that we were seeing with the eye with the fly. And what you can see is that if you look at the pancreas after a week of recovery of this, you get much more scarring in the FicD than you do in the wild type. And this is really interesting because another group came out with this study, infant onset diabetes caused by deregulated ampylation of human endoplasmic reticulum chaperone BiP. And this is this R to S mutant. So as soon as this paper came out, because Amanda is very interested in things that are happening in the pancreas, she said, I want that mice. And she was going to make it. And so the phenotype for the enzyme is it has decreased ampylation, but it has no deampylation. So you're just accumulating inactive BiP inside the ER. And so she went to Jun Wu, who's an excellent geneticist at UT Southwestern. She said, can we make this mouse? And so he made it very efficiently, and so now we have a mouse, R to S. And so I'll just show you a little bit of data, and we'll see what happens with these animals. So basically, if you look at wild type animals, and you look at a low stress, that's a fast. It's a very low stress on your body. But as soon as you start eating, that's a really high stress to your pancreas. And so she's looking at the pancreas, and I want you to look at, is the ampylated BiP under low stress, and then the ampylation goes away under high stress. But then, if you look at these animals, these R to S animals, they have no deampylation, right? So what's happening is they're going to have increasing levels of BiP, ampylated BiP. And so what you can see is that's exactly the case, that you have high levels of ampylated BiP, and it doesn't go away when the ER is stressed. So they have no extra chaperone to deal with this. And so if you look at the animals and ask if they have infancy onset diabetes, you can see that they're glucose intolerant. They're insulin sensitive, so they have the insulin receptors, but if you look at circulating insulin, it's low. And this phenotype is consistent with type 1 diabetes. But this is the coolest experiment that she did. She went back and she looked at the pancreas, and she looked at it in the wild type and the R to S mice. And she says, what do they look like in utero? Because remember, mom takes care of the glucose when you have, basically for the embryos or the POs in utero. I'm not saying the right name. But anyways, before they're born, their pancreas looks fine. So you're looking at their islets, right? And that's where the red arrows are. Sorry about that. But as soon as they're born, she looked at them at 5 weeks, and she's seeing a disorder in the islets of the 5-week-old R to S, and then after a year, it's even worse. And when she looks at this, she doesn't see cell death or inflammation. So this seems like more like type 2 diabetes, where you're getting a de-differentiation of the beta cells in the pancreas. So what I told you today is basically we started out in bacteria, looking at this effector protein, and we kind of followed the science, and we went into flies and mice, and now we've seen it in human disease. And what Amanda has now is this FicRS preclinical model that provides a tool for developing therapies for UPR-induced diabetes, and I think some things are already showing promise for this, as you can imagine with the different tools we have for chaperones. And there's more biochemistry on new and old FIC domains on the horizon. I can tell you more, but I only had this much time. And so, about 15 years ago, doing unbiased basic science, we discovered something very important. FIC encodes a Darwinian trait. Animals survive without FIC, but FIC is conserved and retained in all metazoans because it clearly provides a selective advantage. The FIC rheostat provides resiliency for tissues facing repetitive physiological and environmental stress. And with that, I want to thank my whole lab, and I really, really want to thank Meg for nominating me for this award, and my chair, Eric Olson, Jack Dixon, who first introduced me to pathogenesis, which was just really cool. Joe Sambrook, who took a risk on me, and also to my family, Ron, Ben, and Samantha, for their enormous support. And I'd be happy to take any questions. Thank you.